Here we're going to continue talking about enzyme mechanisms and enzymes and how they function. Again, this comes back to these different chemical compounds that contain carbon can have mirror images, right? They can have enantiomers, which is the mirror image version. And that is when carbon specifically has 4 different and unique things attached. And you can see here the enzyme is this box that this is going to fit into. There's no way that you can rotate this compound and make it fit over here in this, if you have the mirror image, you cannot rotate this compound in any way to make it fit in the same box over here. So these are that a mismatch, they can't fit. No matter how you would want to rotate this around, it's not going to work in here. So stereochemistry is really important for the things that are going to be bound by the enzymes. Here's an example of L-lactate, and it's going to reduce this coenzyme, and lactate itself is going to be oxidized. Lactate dehydrogenase, if you have L-lactate and NAD+, the oxidized form, then you actually have a reduction reaction that happens. But if you have this D-lactate, which is basically this hydroxyl group is switched here, so we have the different form, D-lactate instead of L-lactate, and the oxidized form of NAD, with this enzyme, there's no reaction. And again, it just shows and demonstrates the importance of the substrate and the chirality of that substrate for the chemical reactions to occur. We're going to now go from how enzymes fit onto and attach and recognize the different types of substrates that they react with. And we're going to move into some enzyme kinetics. So understanding how enzymes work and why they work the way they do, and how the energetics of enzymes help to increase the speed of the reactions. Here's an example of a typical type of reaction where we have a nucleophilic substitution. And this is where we're going to substitute one compound for another. And you can see here on this part of the image, we have a negative. Now this is going to be a nucleophile. This is an organic chemistry term. It's a nucleophile because it is negatively charged. It's looking for the nucleus, which we know is positively charged. So something that likes the nucleus, nucleophile. So a negatively charged compound or partially negatively charged compound. Whenever we see these carbonyl groups, we should consider that that is a probable place for a chemical reaction, especially when we're talking about biochemistry. Carbonyl groups are oftentimes involved in chemical reactions. We know that oxygen is partially negatively charged when it's bonded to carbon, and this carbon is going to be partially positively charged. So we have a partial negative charge here, partial positive charge below. This partial positive is going to attract this nucleophile, and the negative electrons here are going to be attracted. They can create this bond. That actually then, since this is negatively charged, we can then flow these electrons towards the oxygen, and we'll get this negatively charged oxygen on the top. Now we've created a new bond between this carbon and this nucleophile. And then these electrons can then flow back towards this carbon, and we can pop off a different subunit. We could, of course, just come back right here and reform this negative Y and just recreate this whole compound, but obviously that wouldn't change anything. When we have a chemical reaction, something changes, and here we're just going to substitute. So it could be these electrons could flow back towards the Y, or they could flow back towards the X. It just depends on which compound is going to be more tightly holding on to those electrons and wanting to share them, or which ones are going to be happier as a negative charge. So here we've substituted this now Y compound for this X, and this was now a substitution reaction. So now we're going to look at the energetics of this. Depending on those types of reaction, there are going to be transition states, and those transition states are going to be different. And the transition state is generally going to be a high energy state. So it's not going to be stable, it's not going to be long-lived, but we have to transition into this less stable this less stable formation before we can get to the products. And here, looking at these energy diagrams, now we can see what actually we can start looking at the reason that the reactions occur the way they do. Here on top is the transition state. So that's that high energy state. When I say high energy, it's right, a lot of free energy. A lot of energy has to go into getting us to this state. So we have to get from this ground state, the compound has to get them to this higher energy state. That state difference between the ground state and the transition state, this is the activation energy, or the energy of activation. This is the amount of energy it takes to get to that point. And now the compound can decide if it's going to go back down to the products or the reactants, or it's going to flow down to the products. And this state, and we'll talk about this a little more in future talks, this is going to be the change in energy. So that's when we're going from, this is kind of when we were talking previously about delta H values. We're going to talk about delta G values as well. Those values, that's describing the energy of the products. So here we started at a higher ground state, we went through this transition state, and we're ending up at a lower state. So there's an overall negative delta value that's going to be associated with this specific type of chemical reaction. You can have simple one-step reactions. You can have multi-step reactions that maybe have two different transition states and it's more stable intermediates, but it has to go through another transition state before we get to the products. So these reaction coordinates are just describing those different steps from one, substrates to transition states to the products, and the amount of energy that are associated with those different states of being. So again, wherever you are on this continuum, you can go forward or backwards on that scale. It just depends on the probability of what's happening with any particular compound at one time. The lower these transition states, if we have a low transition state, there's a higher probability that a compound is going to have enough energy to get up on top of that hump. Really what you have to think about is we have enzymes. They can help change that transition state. They're collecting different types of substrates, right? And then they help go through that transition state and lower the activation energy. So they can put things in the right orientation. They can put everything where it needs to go. And enzymes essentially take that transition state and lower the energy that's required to get to over that hump. And another way of looking at it here, if we look at this uncatalyzed reaction, if we have the enzyme, right, this is the E here, and the two different reactants separately, the enzyme helps bring these together. So it can actually increase, right, that amount of energy that it's starting with once it has these bound. And then the effects of the reaction transition by being bound here, then we can actually lower the activation energy because we can orient them. So collection helps to increase that energy that they have to begin with. And then this next part is kind of more of the orientation. Penmanship is horrible. This says orientation, right? And it orients them so it doesn't take as much energy to get them in the right orientation once they have been collected, and we can reduce the transition state and how much energy is in there. So that means that we need less energy to go from here to here, which means this reaction can happen faster. These are the characteristics of an enzyme that allow it to reduce the size of that intermediate transition state. So again, as I mentioned, proximity, that collection of those things and bringing them in the proper orientation is really important, and that helps to lower the activation energy amount. So we actually want weak product binding as well for an enzyme. So once it has the reactants it bound and in the right orientation, and that chemical reaction occurs, then the product is formed. Well, you don't want to hold on to that product for a long time. You want to be able to release it. So in order for most good enzymes, then so that it can pick up some more reactant, you want to have actually a weak product binding specificity. So it's going to be really high affinity to the reactants, low affinity to the products. Again, that induced fit and that orientation of the substrate is going to be really important. And it should have the ability to stabilize that transition state. We're going to look more closely at this one because this is going to kind of explain how this lowers the activation energy. So that transition state can be stabilized by different intermolecular forces and their interaction with the substrate and the transitioning compound. Before I move forward, I want you to read this question and see if you can answer the question. Otherwise, you should probably go back and do a little more studying here. So we've talked about how these enzymes it's really important that they fit well with their substrate. There are really two models that are used to kind of represent the types of interactions. None of them are perfect or fit every occasion. Historically, the first one was the lock and key model. It came first and described that the substrate is specifically fits into this rigid active site. It's like a key fitting into a lock. But enzymes really are not completely rigid. They're made of proteins. They, depending on that heat or the temperature, they might be a little less rigid and more fluid. And sometimes that's a really important feature. So there's another model, the induced fit model, that accounts for that changing in the shape of the enzyme in the active site to accommodate for the substrate and facilitate the chemical reaction. So there's that idea, and as the enzyme and substrate come together, their interaction helps to induce the right fit for that type of catalysis that needs to happen. Here are some examples of, for binding two different, this is more of a key and fit, but you couldn't get these things together, right? This enzyme couldn't fold around without having a little bit of the ability to move around and be more fluid to cause this induced fit. Again, here's another reaction where there's maybe ionic interactions that are occurring, those intermolecular forces that involve ions to hold that compound in the right orientation and the right place in the enzyme. And then sometimes these aren't perfect fitting things, and there's actually strain put on the molecule, but maybe that strain is really important for helping that transition state and the chemical reaction that's going to occur.
Here we can see a different way of looking at how a binding pocket and a substrate might fit together. And this is called the proximity effect, where an enzyme can trap and collect those substrates in the proper orientation with the help of some of the side chain subunits of the enzyme, the peptide or the amino acids in the enzyme. So for example, here's a serine that might be at the bottom of this pocket. Serine has a hydroxyl group that might be able to interact with this oxygen molecule on the bottom of a tryptophan compound. Aspartic acid down here might also be able to, with its carboxylic acid, be able to interact with the amine groups on the bottom of the arginine. And also the valine right here is just hydrocarbons and might be able to interact with alanine side chains. Threonine has hydrocarbons, but also that hydroxide group, maybe it's able to interact with this amide functional group here on the top end. But this smaller pocket, right, fits this smaller side chain. This larger pocket fits these larger side chains. And then there are support, right? They have the intermolecular forces. That's why I want you to know those. So you can recognize the intermolecular forces that help to bring these in and help to support their insertion into this active site. It helps to promote that proper orientation. Here's another look at a kind of a proximity effect, but some of the mechanism that might happen. And here we have, this is referred to the amino acid side chain, so glutamate, and it's at amino acid 165. So obviously if you have a long chain or a protein of amino acids, this is the 165th one, so you can recognize where it is in the sequence. And so we can see that carboxylic acid of the glutamate is interacting with this hydrogen. So we have this component, this 3-carbon chain with a phosphate group, and we have the ketone here and an alcohol on the side. And it's also interacting with, so we have on both ends, and then we have this histidine 95, so the 95th amino acid of this peptide or this protein is a histidine. This amine functional group is going to interact with that ketone. And so this is kind of the stability, the stabilizing effect, right? So this is the intermediate in order to get this chemical, this compound to essentially turn this ketone into an alcohol, and get rid of that and form a double bond between these 2 carbons, right? That's not a very stable, that's not a very common thing that's happened. But these compounds on the sides, right, they put this thing into the right orientation. They get this so that this oxygen can pull this hydrogen, and this hydrogen can pull that electrons in this oxygen, to allow for that transition state to have a lower energy and actually allow this reaction to move forward when maybe it wouldn't normally happen very quickly outside of this region. I mentioned previously that for the proximity effect to work and for these things to be oriented in the right way, it's really important that we know the types of intermolecular forces that dictate that and help move these compounds in the right orientation. So that's why I required for that organic chemistry component that you know intermolecular forces and which ones are which and how they interact. Because if I don't have these little lines and things drawn for you, I shouldn't have to write that on there. You should be able to recognize, oh, that's a dipole-dipole, that's a hydrogen bond, that's an ionic interaction that's going to happen there. So that's what I'm moving towards. I'd like you to be able to start doing. Obviously, the binding pocket is going to recruit the substrate. But what about the product? What happens after that substrate has transformed in that enzyme pocket? Then what happens? So the question is, should an enzyme have a higher affinity for the substrate or the product, and why? So think about this, come up with a thought on that, and then resume this presentation. Well, the answer to this is that really we want to have a really high affinity for the substrate, but once that transition state happens and the product is formed, we really need that enzyme then to release this. Right? We want to release that product so that it's not bound and trapped in here. This pocket is important for binding substrates and converting them into enzymes. And if it's full of and converting them into products. And if it's full of product, it can't bind to another substrate. So a good enzyme gets rid of this pretty quickly and has a fairly low affinity for that final product. A lot of times these binding pockets will move and they'll change conformation. So when they're bound to something, they have one conformation, and then once this chemical reaction occurs, they'll change their conformation. It'll move how everything's fit together, and that makes this enzyme maybe non-reactive and a less high affinity for that now substrate that's a new product, and it'll eject that so that it can start over again and find something else to interact with. This is a 3-dimensional shape of hexokinase. So it's an enzyme that's involved in glycolysis, and I just want to show you the difference here in what can happen. Oops, sorry about that. So once the substrate's in there, it binds and moves. So that's that induced fit kind of model that I'm showing you here. The ability for this to kind of chomp down and get these different components, right? These side chains now are able to better interact and manipulate that substrate. Here's just a image of a transition state stabilization and how it works with the different intermolecular forces that can be involved. In the middle here we have adenosine. Here's the kind of the base part of the nucleic acid, and here's the sugar component of the nucleic acid. And this on the outside right here, histidine 14, asparagine 16, right? So we have these glycine 8181. These are on that protein chain, the different amino acids on that and where they are located. You'll notice they're not all linear, right? So we have 14, 16, 569, 181, 212. These are all folded up in different ways, and it just happens to be that this binding pocket is made up of these. So this is adenosine deaminase, are those amino acid side chains, and the adenosine is here in the middle. So here you can notice that the hydrogen bonding between the asparagine 292, the adenosine group. You also notice that oxygen and that zinc holding in this zinc molecule.
This is also called a cofactor.
Something else that we're going to talk about, vitamins and minerals are sometimes really important in stabilizing these active sites. So we have different metals in our bodies that enzymes bind to, and they use those enzymes to help with some of these transition states and stabilizing the transition states of enzymes. Both the cofactors and coenzymes do this. So I just wanted to point that out. That's really their role. That's what they do. They're just used for enzymes for these transition state and for proper folding of that components, right? It's not only stabilizing this asparagine, right, and this histidine and these histidines. It's helping orient and organize and properly fold that enzyme, and it's also being used in this active site for this transition state. So they have a lot of times multiple roles in both helping fold that protein and then part of the active site or part of the function of that protein. So we've been so far looking mostly at enzyme substrate interactions or transition state interactions with the enzyme and the substrate. But here's kind of the whole thing in action and the different steps that are involved. So here is the ES is the enzyme substrate component complex. Here's the transition state complex in the middle, and then the enzyme product or EP complex at the very end. The substrate we're looking at is an oligosaccharide, which is 6-sugar component. Only the D and E sugars of that component are shown here, just for clarity, and they're down in the middle here. So here's the pocket, and here's glutamate 35, so it's the 35th amino acid on this protein backbone, aspartate 52, 52nd amino acid. So we have both that glutamate, the hydrogen bonding interaction with that carbon to oxygen backbone of that sugar molecule, and this negatively charged oxygen here is going to be attracted to that partially positive carbon, right? Those carbon electrons are being pulled towards these oxygens, so there's going to be a partial positive charge, which is going to be attractive for that negative charge on the oxygen. So this is what holds this in place and stabilizes that initial interaction here. I'm sure there are multiple other regions here, but we're just focusing on this region right here, which is going to have undergo an enzymatic chemical reaction, right? Here we have water, so we see that this is what type of chemical reaction? It's going to be hydrolysis, right? So this glutamate also helps to, once it's donated that hydrogen, to put that in place, right? This hydrogen gets donated, this oxygen grabs it, electrons move from this carbon-oxygen bond to that hydrogen so that we can form this hydrogen bond. This water comes in and it's now able to be added to, now this is the transition state, so we have this temporary bond from aspartate 52 with this sugar backbone, but we have to reform. That's what enzymes do and catalysts do. They reform their original shape. In order to do that, we have water come in and rehydrate or reduce that oxygen here, add a hydrogen. This hydroxide gets added to that carbon end, so we release that aspartic acid, and we reform that aspartic acid, reform that glutamate, and now we have used hydrolysis really in the end, right? We've added a water molecule to the end to break this sugar backbone chain. So both of these, we're using carboxylic acid side chains. One we have initially reduced, one we have oxidized, but we have to recycle that in the very end to reform that.
Now we're going to have a very low affinity to this new product because right before we were interacting with that backbone and that carbon, and now those are kind of bound with other things. So there's going to be a low affinity. This substrate is going to be ejected out of this site pretty quickly. Here's another example of an enzyme in action, and this is in blue, the binding pocket of the active site of chymotrypsin. So you'll notice that the side chains, serine 195, histidine 57, aspartic acid 102, right? These are some of the important components of that amino acid that are in that binding pocket that are going to actually be involved in the hydrolysis. Remember, hydrolysis is the breaking of bonds and adding water to help break those bonds. And it's an enzyme that breaks apart amino acid peptide bonds. So we're going to break apart this amide bond. Phenylalanine is going to be recognized by the hydrophobic pocket. So this pocket probably contains amino acids in this protein. They're basically hydrocarbons. They're going to be valine, alanines, different non-polar side chains that are going to help bind to this non-polar phenylalanine benzene ring. So we'll have that binding pocket that helps to orient this thing in the right place and helps to get this in position to set up this amide bond to help histidine and serine help cleave that. You can see that then this orientation, we have the serine, this hydroxyl group actually makes a temporary covalent bond with this carbon that's attached now to this bond, and we've helped to pull this hydrogen off with this histidine. Nitrogen, the amide helps to accept that hydrogen. Remember, these hydroxides, these alcohols are generally not very acidic, but it helps if there's a base here. And so this all is helping to create this system where we can have this chemical reaction. So this hydroxide negative charge can then attach to this partially positive carbon. And now what we can do is we can actually have this hydrogen attached to this nitrogen, and then it will release this carbon, and we've broken and created that free peptide. So again, these enzymes catalyze this in 4 ways. We bring it in, we bring these substrates together, and we put this in, right, orient it in the proper way by having this kind of stuck into this hydrophobic pocket. There's a catalytic effect because we have acids and bases and the proper things that allow, maybe this doesn't happen in a high degree if it's floating out by itself in a solution, but in this environment, this alcohol is able to release, it's able to act kind of like an acid and release that hydrogen. We get a really negatively charged oxygen, so it's going to be really attracted to that very close and in proximity carbon, partially positive carbon, and then we're able to start that chemical reaction, right? We're able to make this intermediate that's normally very difficult to make, but because of how this is in the proximally close, it's oriented in the right place, and we have the right things in the right spot, right? We have the catalytic effect, and that ultimately lowers the energy barrier. So the energy effect is that intermediate is stabilized because of all of these things that we just talked about. Those different mechanisms now lead us into thinking about enzyme kinetics, and a guy called Michaelis Menten who developed this idea about thinking about enzymes and how that enzymes change the speed at which a chemical reaction occurs. So we have been talking about enzymes and their substrates, and that intermediate, that enzyme substrate intermediate, takes a certain amount of time to form, and that can be represented by this K1 value. So that's how fast this happens is represented by the enzyme constant 1, and the falling apart back into the enzyme substrate is the K -1, right? It's just telling us this -1 is telling us it's the reverse reaction. Enzyme substrate can also fall apart into the enzyme and the product, and that can be described by a certain speed at which that occurs would be the K2 value. Obviously, coming backwards would be K -2 as well. Here is the way that we actually represent what those K values mean, actually, those constants. What is that telling us? And it's really talking about the velocity of the chemical reaction. And what you'll notice here is how we can generally look at this. We can look at the substrate or the product, the change over time, right? That's what delta means, is change of the concentration of the product over the amount of time it takes to change that. So we're looking at molarity of product versus how that changes over time. We can look at substrate as well. A lot of times we're going to think about that constant value times the amount of substrate. The substrate's critical because that's what the enzyme's recognizing. The concentration of the substrate is oftentimes related to how fast the chemical reaction is occurring, not the concentration of the product. Even though the product is what maybe we're interested in looking at, how fast that's formed, it's really the substrate concentration that's going to dictate how fast things happen, because the substrate's essential, because that's, again, what the enzyme's binding to. I want to also just briefly introduce some topics, and we're going to talk about this in more detail in a little bit. But this is really the graphing of a typical enzyme catalyzed reaction. A couple things that you should know. One of the big things is the maximum velocity. So if we have as much substrate as we possibly can, so this graph we're looking at the velocity, how fast that reaction is consuming substrate and turning it into product, and that this is related to the amount of substrate. So the more substrate we add, the more fast that reaction can go. But you'll notice that there's once this we add more substrate, we get more speed, we add more substrate, we get more speed, but it dies off eventually. So the speed, although it's dictated by the substrate concentration, there's a maximum speed that ever is obtained. We can only have so much substrate in there. There's only so much enzyme that can act, and so that speed is dictated by the amount it gets saturated at some point. So this is the maximum velocity. This is one important term, Vmax. At half Vmax, half of the maximum velocity, if we would go over to this region, we can find the spot that's on this curve and go down to this concentration. This is a Km value. This Km value is the substrate concentration at half Vmax. What it tells us, the different curves are going to have different Km values, right? If this curve is less steep or more steep, that Km value is going to be different depending on that different type of enzyme. And what it does is it tells us something about how good that enzyme is at binding to the substrate. This Km tells us really affinity. Tells us about how good, and affinity is equal to how good it binds. And what you'll notice as you look over here, right, the better it binds, the faster the speed is at a lower substrate concentration. So the lower the Km value, higher affinity. And that is because, again, if we're looking at this chart, the lower the Km value, it means it's going faster at a lower substrate concentration. Km, and that tells us that enzyme's really good at finding and binding that substrate. So a couple of important terms, that Vmax and Km, and we'll come back to that. But I just wanted to introduce that to you right now. And what we're going to do in class, we have an in-class activity looking at things. And what you should think about, what are some things that you think might influence enzyme activity? We've kind of discussed them a little bit, talking about digestion, right? You could be able to think about how the difference is in physiologically as things go through our bodies, how that changes, what we've done in maybe the lab a little bit. What things do you think that can modify enzyme activity?